In this lecture, I will talk about lectin complement pathway. In my previous class, I have already discussed about classical complement pathway. So let's start. So here also some complement proteins take part in this pathway, like C2 to C9 proteins that are also responsible for classical complement pathway. So in this lectin pathway, there is also C2 to C9 complement proteins are involved. But there is a difference between lectin pathway and the classical complement pathway, the initiator protein. In case of classical complement pathway, the initiator protein is C1q protein. But in case of lectin pathway, the initiator protein is MBL. It means Mannose binding lectin. Lectin means carbohydrate and Mannose means it is a sugar. So MBL means Mannose binding lectin, and very recently it is discovered that another protein called ficolin is also acts as a initiator protein in lectin pathway. So this MBL and ficolin, the binding ligand of these two molecules are MBL, Mannose binding lectin, can bind with Mannose sugar that are present in the cell wall of bacteria, fucose sugar, high affinity with Mannose and fucose, and the ficolin can bind with acetylated oligosaccharide. It's like N-acetyl glucosamine. So if I draw a bacteria, so this bacteria have the cell wall. In this cell wall, these are the Mannose, fucose and acetylated carbohydrate. So Mannose, fucose and acetylated oligosaccharide that are present on the cell wall of bacteria. If any bacteria enter our bloodstream, the blood serum protein MBL and ficolin can recognize the cell wall Mannose, fucose and acetylated oligosaccharide, so they can bind. This MBL, Mannose binding lectin, and the ficolin and C1q protein, all the initiator protein of lectin pathway and the classical pathway share very common structure. They all have 6 arms. Now come to the reaction part of this complement pathway. So when a bacteria enter into the bloodstream, blood serum contain MBL or ficolin that can recognize this ligand. It's actually initiator protein. The MBL and the ficolin have 3 serine proteases that are called MASP1 to 3. So MASP1, MASP2 and MASP3. The full form of MASP means MBL associated serine protease. Especially the role of MASP2 and the structural similarity of MASP2 protein, it was found it is very similar to C1S, 1S of the complement protein of classical pathway. So it works as a serine protease. So I can write it MASP2 mainly, perform the enzymatic function MASP2. What happens? The MASP2 actually cleave 2 complement protein. One is C4 and another is C2 into its 2 components or fragment. In my previous class, as I have already mentioned, the complement proteins, when get cleaved, it produce 2 fragments. One is called C4A, A for smaller fragment, and C4B for larger fragment. Also the C2 will cleave to and produce C2A, C2B. But here is a difference and exception that in all the complement protein like C4, C3 and C5 follow the B rule, means B is equals to larger fragment and A is equals to smaller fragment. These 3 molecules C3, C4 and C5 follow the B is equals to larger and A is equals to smaller fragment rule. But there is one exception, that is C2. C2 when cleaved, they produce C3A and C3B. Here C3A is the larger fragment and C3B is the smaller fragment. So what happens when the MASP2 cleave this C4 and C2 molecule? This molecule gets cleaved into 4 fragments, C4A, C4B, C2A, C2B. Now, as these molecules are larger fragment C4B and C2A, they join each other and form an enzyme called C3 convertase. So now these two larger fragments attach with each other form C3 convertase, and in my previous class I have mentioned that C4A, the smaller fragments of complement protein, acts as a anaphylatoxin. Anaphylatoxin means that chemical that can induce inflammation. As the name implies, the C3 convertase can cleave C3 complement protein. So it can cleave the C3 protein into two fragments and produce C3A and C3B. Now C3A is the smaller fragment and C3B is the larger fragment. Also, the C3A function as a anaphylatoxin. So you can see both the smaller and the larger fragments have its own function. This C3B protein can join with this complex C4B C2A. So now this complex, that is C4B attaching with C2A, now join with this molecule to form C3B, a complex. So now you can see this C3B join with this molecule to form another complex called C4B, C2A and C3B fragments. This is called C5 convertase. I want to mention in this case that this C3B protein also acts as a opsonin. So the ultimate product is C5 convertase in this reaction. Now C4B, C2A and C3B, the C3 convertase cleave C5 into two fragments. One is C5A and C5B. Here is also the C5A acts as a anaphylatoxin and the C5B now released into the serum. After forming C5B, the C5B protein attach with C6. Then the C5B and C6 can bind into the bacteria. Now this complex protein can bind with the bacteria, and after binding, C7 are recruited. After C7, C8 protein are recruited, and then C9 protein are recruited, and the multiple of C9 protein form a molecule called MAC, that means membrane attack complex. Okay, so this MAC is actually made up of many C9 proteins that form a pole-like structure into the bacteria, and by the way, the bacteria actually die.